An introduction to the fungi for microbiology. At least 4 properties distinguish fungi from plants. Fungi lack chlorophyll, while plants have this pigment. Fungal cell walls contain a carbohydrate called chitin. Most fungi are not truly multicellular like plants. Fungi are heterotrophic, while plants are autotrophic. Fungi are placed in their own kingdom, fungi, with the domain Eukarya of the tree of life. The study of fungi is called mycology, and a person who studies fungi is a mycologist. Fungi generally have life cycles involving 2 phases, a growth phase or vegetative phase and a reproductive phase. Molds grow as long, tangled filaments of cells that give rise to visible colonies, while yeasts are unicellular organisms whose colonies on agar can visually resemble bacterial colonies. Molds consist of masses intertwined with filaments called hyphae. The hyphae are the morphological units of a filamentous fungus. The thick mass of hyphae is called a mycelium. Fungi have 1 or more nuclei, as well as a range of organelles, including mitochondria, an endomembrane system, ribosomes, and a cytoskeleton. The cell wall is composed of large amounts of chitin. Chitin is a carbohydrate polymer of acetylglucosamine units, that is, glucose molecules containing amino and acetyl groups. In many species of fungi, hyphal crosswalls, called septa, divide the cytoplasm into separate cells. Such fungi are described as septate. If the septa is absent or incomplete, such fungi will be described as coenocytic, meaning they contain many nuclei in a common cytoplasm. Because fungi absorb preformed organic matter, they are described as heterotrophs. Many fungi are known as saprobes that feed on dead or decaying organic matter. Together with many bacterial species, these fungi make up the decomposers. Other fungi are pathogenic and live on plants or animals, causing disease. Most such fungi are opportunistic and only cause serious disease in immunocompromised individuals. Fungal growth is dependent on moisture, adequate chemical nutrients, and physical factors. Molds and yeast secrete enzymes into the surrounding environment that break down or hydrolyze complex organic compounds into simpler ones. The mycelium formed by the molds represent a feeding network for the fungi. In some cases, the mycelium can form a tremendously large surface area for nutrient absorption. Yeast growth in number occurs by cellular division, as cells undergo many rounds of mitosis and cytokinesis to form a large population of individual cells. The availability of chemical nutrients, oxygen, temperature, and pH influence fungal growth. The majority of fungi are aerobic organisms, with the notable exception of facultative yeasts, which can grow in either the presence of oxygen or under fermentation conditions. Most fungi grow best at about 23 degrees Celsius, a temperature close to room temperature. Notable exceptions are pathogenic fungi, which grow optimally at 37 degrees Celsius, which is body temperature. Psychrophilic fungi grow at temperatures such as 5 degrees Celsius, which is normal refrigerator temperature. Many fungi thrive under mildly acidic conditions at a pH between 5 and 6. Acidic soil, therefore, may favor fungal turf diseases, in which case lime or calcium carbonate is added to neutralize the soil. Mold contamination also is common in acidic foods, such as sour cream, citrus fruits, yogurt, and most vegetables. Moreover, the acidity in breads and cheese also encourages fungal growth. Normally, high concentrations of sugar are conducive to growth, and laboratory media for fungi usually contain extra glucose. Fungal growth in nature forms important links in ecological cycles because fungi, along with bacterial species, rapidly decompose animal and plant matter. Working in immense numbers, Fungi release carbon and minerals back to the environment, making them available for recycling. Many fungi also live in mutually beneficial relationships with other species in nature through a symbiotic association called mutualism. Mycorrhiza consume some of the carbohydrates produced by plants, but in return act as a second root system, contributing essential minerals and water to promote the plant's growth. Sporulation is the process of spore formation. Sporulation usually occurs in structures called fruiting bodies, which represent the part of a fungus in which spores are formed and from which they are released. These structures may be asexual and invisible to the naked eye, or they may be sexual structures that are macroscopic and visible to the naked eye, such as in mushrooms. Asexual reproductive structures develop at the ends of specialized hyphae. As a result of mitotic divisions, thousands of spores are produced, all genetically identical. Many asexual spores develop within sacs or vessels called sporangia. Appropriately, the spores are called sporangiospores. Many yeasts reproduce asexually by a process called budding. In this process, the cell becomes swollen at one end, and the new cell, called a blastosphere, develops or buds from the parent cell. Eventually, the spore breaks free to live independently. The parent cell can continue to produce additional blastospores. Once free of the fruiting body, spores landing in appropriate environments then will have the capability of germination, reproducing new unicellular yeast cells or a new hypha. Continued growth will eventually form a mycelium. Many fungi also produce spores by sexual reproduction. In this process, opposite mating types come together and fuse. Because the nuclei are genetically different in each mating type, the fusion cell represents a heterokaryon, that is, a cell with genetically dissimilar nuclei existing for some length of time in a common dikaryotic cytoplasm. Eventually, the nuclei fuse, and a diploid cell is then formed. The chromosome number soon is halved by meiosis, returning the cell or organism to a haploid condition. A visible fruiting body often results during sexual reproduction, and it is the location of the haploid spores. Perhaps the most recognized fruiting body from which spores are produced is the mushroom. Let's look at the typical life cycle of a mold. Many fungi have both sexual and asexual reproduction characterizations. Reproduction characterized by spore formation. The unique phase in the life cycle is the presence of the heterokaryon, where nuclei from two different mating types remain separate or dikaryotic in a common cytoplasm. A fungus can be cataloged into one of 5 phyla, depending on its mode of sexual reproduction. These phyla are Chytridiomycota, Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota. If the fungus lacks a recognizable sexual cycle, it is placed into an informal group called the mitosporic fungi. The oldest known fungi are related to certain members of the Chytridiomycota, commonly called the chytrids. Members of this phylum gives us clues about the possible origin of fungi. Chytrids are predominantly aquatic and not terrestrial organisms. This means the fungi originated in water, probably from a flagellated protistan ancestor. Secondly, being aquatic, chytrids have flagellated reproductive cells. No other fungi have motile flagellate cells, suggesting other fungi may have lost this trait at some point in their evolutionary history. Finally, like no other fungi, the chytrids have chitin strengthening their cell walls. The Glomeromycota form what most consider the most extensive symbiosis on Earth. These fungi represent a group of endomycorrhizae that exist within the roots of more than 80% of the world's land plants. They do not kill the plants, but rather they interact mutualistically by providing essential phosphate and other nutrients to the plant. In return, the fungi receive needed organic compounds from the plant. In fact, some mycologists believe that plant evolution onto land more than 400 million years ago depended on the symbiosis of the ancestral Glomeromycota, which were able to provide plants with the needed nutrients from the soil. The phylum Zygomycota consists of a group of fungi inhabiting terrestrial environments. Familiar representatives include the fast-growing bread molds and other molds that typically grow on spoiled fruits with a higher sugar content or on acidic vegetables. Members of the phyla Ascomycota are very diverse and account for 75% of all known fungi. The phyla contains many common and useful fungi, including Saccharomyces cerevisiae, baker's yeast, Morchella esculentum, the edible morel, and Penicillium chrysogenum, the mold that produces penicillin. The Ascomycota phylum also has several members associated with illness and disease. Aspergillus flavus aflatoxin, which is a fungal contaminant of nuts and stored grain that is both toxic and carcinogenic. Cryphonectria parasitica is responsible for the death of 4 billion chestnut trees in the eastern United States. Candida albicans is the cause of thrush, diaper rash, and vaginitis. Members of the phylum Basidiomycota are club fungi. The Basidiomycota contains about 30,000 identified species, representing 37% of the known species of true fungi. This phyla includes the mushrooms. Let's look at the sexual reproduction of a mushroom. On the underside of the mushroom cap, the channels or gills are lined with club-shaped sexual reproductive structures called basidia. Nuclear fusions within the basidium give rise to the zygote, which is a diploid cell. The zygote then undergoes meiosis, and haploid basidiospores form. Basidiospores are released from basidia, and they germinate to form hyphae of different mating types. Growth results in a haploid mycelium. Hyphae of opposite mating type unite and form a heterokaryotic mycelium. The hyphae of the heterokaryotic mycelium emerge from the soil as a mushroom. Basidia form on the underside of the cap to complete the cycle. Certain fungi lack a known sexual cycle of reproduction. Consequently, they are labeled with the term mitosporic fungi because the asexual spores are the product of mitosis. The word yeast refers to a large variety of unicellular fungi. Some of these yeasts include Saccharomyces, which is the fungi that has the ability to ferment sugars. Most of the best known yeasts are Saccharomyces. The Saccharomyces species S. cerevisiae are used for bread baking and are known as baker's yeast. Saccharomyces species Ellipsodius are used for alcohol production. Although there are no major infectious diseases of the digestive system caused by fungi, some mushrooms, molds, and yeast produce toxins called mycotoxins. Although these toxins are ingested, they may have poisonous effects that reach far outside the digestive system. Two closely related fungi, Aspergillus flavus and A. parasiticus, produce mycotoxins called aflatoxins. Aflatoxins are deposited in foods and are ingested by humans where they are thought to be carcinogenic, especially in the liver. Ergotism is caused by Claviceps purpurea, ascomycete fungus, that produces a powerful toxin. C. purpurea grows as hyphae on kernels of rye, wheat, and barley. As hyphae penetrate the plant, the fungal cells gradually consume the substance of the grain, and the dense tissue hardens into purple bodies called sclerotia. A group of peptide derivatives called alkaloids are produced by the sclerotium and are deposited in the grain as a substance called ergot. Products such as bread made from rye grain may cause ergot rye disease, also known as ergotism. Symptoms may include numbness, hot and cold sensations, convulsions with epileptic-type seizures, and paralysis of nerve endings. Lysergic acid diethylamide, or LSD, is a derivative of an alkaloid in ergot. Commercial derivatives of these alkaloids are used to cause contractions of the smooth muscle, such as to induce labor and relieve migraine headaches. Some mushrooms produce mycotoxins. A poisonous mushroom refers to any mushroom that produces various toxins that can cause an adverse or harmful reaction when eaten. By this definition, of the more than 5,000 species of identified mushrooms in the United States, about 100 are responsible for mushroom poisoning, but less than a dozen species are considered deadly. Mushroom poisoning, or mycetism, can occur from mushrooms that produce mycotoxins that affect the human body. Some are neurotoxins that may 1. Mimic the action of acetylcholine, affecting the peripheral parasympathetic nervous system. 2. Generate psychotropic effects or hallucinations. Or 3. Produce a condition resembling alcohol intoxication. Deaths are rare, but in severe cases, the intoxication may cause cardiac or respiratory failure. Further, in children, accidental consumption of large quantities of some mushrooms may cause convulsions, coma, and other neurological problems for up to 12 hours. Amanita phalloides is referred to as the death cap because it accounts for about 90% of deaths from eating poisonous mushrooms. About 10 to 14 hours after ingesting just 1 to 3 mushrooms, enough of the protein toxins, called amatoxins and phylotoxins, are present in the body to produce gastrointestinal symptoms of severe abdominal cramping, vomiting, and watery diarrhea. The symptoms last for about 24 hours and can cause dehydration and low blood pressure. In humans, fungal diseases, generically known as mycoses, often affect many body regions. Dermatophytosis is a general name for fungal diseases affecting the hair, skin, or nails, commonly known as tinea infections, tinea meaning worm, because in ancient times worms were thought to be the cause of these diseases. Tinea infections compose several forms of ringworm, including athlete's foot, known as tinea pedis, head ringworm, known as tinea capitis, body ringworm, known as tinea corporis, groin ringworm, or jock itch, known as tinea cruris, and nail ringworm, known as tinea unguium. Candida albicans often is present in the skin, mouth, vagina, and intestinal tract of healthy humans, where it lives without causing disease. The organism is a small mitosporic yeast that forms filaments called pseudohyphae when cultivated in laboratory media. When immune system defenses are compromised or when changes occur to the normal microbiota of the body, C. albicans can flourish, causing numerous forms of candidiasis. One form of candidiasis occurs in the vagina and is often referred to as vulvovaginitis or a yeast infection. People who work with wood products, or soil can contract a condition called sporotrichosis. Sporotrichosis is an infection caused by Conidia sporothrix schenckii. People may come in contact with this by handling sphagnum or peat moss that is used to pack tree seedlings, or it can be transmitted through skin punctures like those from rose thorns. Sporotrichosis can result in pus-filled purplish lesions that form at the site of entry and form knots that may be felt under the skin. Cryptococcus neoformans causes a rare but dangerous respiratory disease called cryptococcosis. Cryptococcosis is among the most dangerous fungal diseases in humans. It affects the lungs and meninges and is estimated to account for over 25% of all deaths from fungal disease. This condition is caused by an oval-shaped yeast called Cryptococcus and is a member of the Basidiomycota. The organism is found in the soil of urban environments and grows actively in the droppings of pigeons, but not within pigeon tissues. Cryptococci may become airborne with gusts of wind, and the organisms subsequently enter the respiratory passageways of humans. Histoplasmosis is a lung disease most prevalent in the Ohio and Mississippi River valleys, where it is often called the summer flu. The causative agent is Histoplasma capsulatum. The lungs are the primary portal of entry. Thus, infection usually occurs from the inhalation of spores present in dry, dusty soil or found in the air of chicken coops and bat caves. Blastomycosis occurs in Canada, the Great Lakes region, and areas of the United States, from the Mississippi River to the Carolinas. Acute blastomycosis is associated with dusty soil and bird droppings, particularly in moist soils near barns and sheds. Inhalation leads to lung lesions with persistent cough and chest pains. Entry to the body also may occur through cuts and abrasions. Symptoms include raised wart-like lesions that may be located on the face, legs, or hands. Travelers to the San Joaquin Valley area of California and dry regions of the southwestern United States may contract a fungal disease known as coccidioidomycosis, known more commonly as valley fever or desert fever. It is caused by Coccidioides immitis in California or Coccidioides posadaceae in Arizona. Coccidioidomycosis is usually transmitted by dust particles laden with fungal spores. Cattle, sheep, and other animals deposit the spores in soil, and they become airborne with gusts of wind. When inhaled into the human lungs, C. immitis induces, within 7 to 28 days, an influenza-like disease with a dry, hacking cough, chest pains, and a high fever. Let's look at the life cycle of Coccidioides immitis. Outside the body, this organism exists as a septate mycelium. It fragments into airborne arthrospores, which are inhaled. In the respiratory tract, these arthrospores swell to yield a large body called a spherule. The spherule segments and breaks down to release endospores. When released to the environment, the endospores form germ tubes, which then mature, forming the mycelium. Pneumocystis pneumonia, or PCP, currently is the most common cause of non-bacterial pneumonia in the Americas among people that have suppressed immune systems. The causative organism is Pneumocystis jirovecii, which produces an atypical pneumonia that remained in relative obscurity until the 1980s, when it was recognized as the cause of death in over 50% of patients dying from the effect of AIDS. Thank you for watching.